Today we're featuring the Harvard MIT Neuroimaging Program, and this is our pilot seminar, which we hope to turn these into a seminar series, inviting all of our T32s to take turns, come and introduce themselves. So whatever feedback you can provide us on this pilot will be really helpful for us, both on the side of the audience and as well as speakers, like what they may have like to see differently in the future about format or content or anything. I want to mention that we're recording the session and hope to make it online. And before I turn things over to Randy, I wanted to just ask people to turn on their cameras if they're not terribly opposed to it, so that we can see each other and maybe take a snapshot. And then you can turn it off again later if you don't like to be on. Anybody more? Any takers? Okay. And then I will pass it on to Randy. And I just want to say that Randy is the co-director of the Harvard MIT Neuroimaging Program. And I've had the pleasure of working with her on arranging this series, working out the details. It's been a real joy, as it always is working on other things with Randy. And I just want to add that she's a professor of psychiatry at Harvard Medical School and Mass General Hospital, and holds a secondary appointment at radiology in MGH. Randy?
Well, thank you for that lovely and kind introduction. It is really a pleasure and an honor to be here. So I'm really, I was so glad when it worked out that we could do this with you together. And so I am going to serve as sort of MC for our team presentation to you today. I want everybody to know that there is a chat you can put questions into if you have them, or you can just unmute and interrupt us. And because all of us like taking questions and we like this dialogue and discussion, we are a very talk to-ish kind of people where we are. And also available to anyone for questions or follow-up after the presentation's over. We'll leave you our contact information. So thank you for having us. I am going to share slides because I just love brain imaging. There's just gorgeous things to share and show. And so I will be using slides, but I want to point out one thing, that our training program has a nice website. So any questions that don't get answered here, you can Google us and find us easily. And also our contact information is here. But today I'm going to tell you a little bit about our program, the sort of big picture kinds of things. And then I'm going to pass the baton to my brilliant co-director, Bruce Rosen, who is going to give you a little taste of the kind of science that our students do from the big picture, from the faculty side. And then you're going to hear from 3 of our current trainees at different stages in the program. And they'll tell you a little bit about what their experiences are like, because that's probably the most relevant and interesting for you. So let me just make sure yep. So we've been around a while. The NTP was started in 2003. I had our first class, and so really proud that some of the very first of our trainees that we supported are now faculty teaching training our new students, which is very cool. And we're getting almost old enough to have children of our children becoming faculty. We're getting close to that. So how do you become a student in this program? So it's after you graduate, you're applying for graduate schools. You apply through the regular application process at our home organization, home program. It's the Harvard MIT Medical Engineering and Medical Physics program. We call it MEMP for short. to be part of our neuroimaging training program cohort of people who will benefit from what this grant enables us to do, you just have to self-identify that neuroimaging is interesting to you, and then you're welcomed and you're on our email list and invited to all of the activities and things that we do together. And you're also then eligible to apply for the funding slots that are available for a couple of the years, 1 or 2 of the years that you are in the program, which we're very grateful for. It helps out our faculty a tremendous amount to have that. What is unique about our PhD program is that all of our trainees get a very deep introduction to clinical medicine as compared to many of the other biomedical engineering type programs or engineering type programs. So they actually do a introduction to clinical medicine rotation where they are in hospitals, get to see patients, get to, a real in-depth experience, and also get to do an elective in something that's very specifically interesting to them, working with a neuroradiologist or an interventional radiologist or something like that, for example, for our program. There are, of course, the required classes and thesis research that is standard to most graduate programs. But what sets our program, what Zeynep asked me to let you know, is really important for our program that people have some research experience when they apply. So a lot of our students have not only graduated from college, they may have already done some research during their undergraduate years, and most of them have. They also often will take a year or more off between then and applying and do more research, get more experience, have more skills that they then bring when they start the program. That is not an atypical path, but you'll hear a couple of stories along those lines. Of the kinds of fun things that the NTP program allows us to do is we have some special luncheons where we help students to get introductions to things like non-academic careers and interview people whose paths might be different from what they would know about. So they get introduced to more different kinds of things that they do. We also work with them to put on a symposium every other year on one of the topics that's near and dear to our hearts. The one we just did on mesoscale mapping was in partnership with Bruce Rosen's P41 grant. And so these featured speakers, most of them are faculty of the P41, and many of the trainees supported by the NTP were presenters presenting their posters in the poster session. And now those students are working with me and the faculty to write up the results, a review article on this topic for publication. And it's a really great learning experience. It gives a much more in-depth connection to this new material for them. And just a quick highlight that we have a lot of faculty mentors, tons of them. They're amazing, each one in their own right. Lauren O'Donnell is actually the example of was in the class of first trainees in 2003. She's now associate professor and she's been on the grant as faculty for a while. But we have neurosurgeons. Alex Goldby is a brilliant neurosurgeon. Ron Kakinis, a brilliant medical image analysis. And I put a URL there for a YouTube video so you can get a really exciting view of the intraoperative imaging suite that is over at the Brigham. Our facilities are unparalleled. The number of places that you can work from our program across all the different Harvard-affiliated teaching hospitals is really breathtaking. There's so many opportunities. If you're interested more in early life applications, Ellen Grant, another brilliant faculty member of ours, is running a big research program over at Boston Children's Hospital, and they multimodal imaging, state-of-the-art facilities and amazing applications. And our newest joining us in our competing renewal application will be Yangming Wu, who's now up for made associate professor recently. Ellen and I co-mentored him during his postdoctoral year. So really a great program. I want to make a quick shout out to a project that is up and running at our center that might be of particular interest. It's this Laboratory for Computational Neuroscience has a collaborative internship, a summer internship for high school students. And it has a beautiful program, a summer program, where these high school students who might otherwise not have much exposure to STEM can come in and actually do research on neuroimaging, have very close mentorship and a lot of professional development help so that they can make the step from high school to college. And this program is funded through NIH. Thank you very much for this as well. And it's been really a lovely program. It works in close collaboration with a program called My Brother's Keeper for bringing in underprivileged students who might not have another opportunity to have an experience like this. And they recruit great students to come in and help. We reach broadly to make mentors out of all of our research coordinators and our junior scientists. And a thought that I had is that if any of the esteemed scholars wanted to do a summer internship with us, you guys could be the mentors to these high school students who are there for the summer. It would be kind of like mentor the mentors. Your name could be on our list of mentors or form sort of a mentorship team. So part of learning to be a good scientist and a good teacher, you have to get experience. And so this might be a really wonderful opportunity. So we welcome you to think about that. So now I'm gonna pause and turn the mic over to my colleague, Bruce.
Thanks, Randy. That's great. And nice to meet all of you. I will say I was a graduate of that medical engineering, medical physics program myself sometime just before the Earth cooled. But it's a fantastic program. Lots of great things. Of course, neuroimaging is the focus of this program, but even if you have no interest in neuroimaging, I would absolutely encourage you to check out the MEM program. It's this really amazing opportunity, as Randy said, to kind of be taking classes with medical students, getting in the hospital, but then also getting all the rigorous engineering and electrical engineering, computer science or mechanical engineering or physics or chemistry, all the benefits of the rigorous engineering and physical sciences, coupled with, an exposure to the world of medicine that's hard to match in other places. So Randy has teed me up to just highlight just a few examples of the kind of things that we do here at the Martino Center, and that are, of course, opportunities for thesis research for our undergraduates and especially graduate students. One is something that we call the Connectome 2.0. You might guess from the title that there was a Connectome 1.0, right? There was an original Connectome. This is a program that's led by Susie Huang, who is a MD, PhD radiologist and physicist within the Martino Center. About a decade ago, she set out with colleagues to build a scanner that was really kind of a one of a kind scanner designed to image how the brain is wired, the human connectome. 10 years on, it was time to kind of improve upon that, and that led to a grant, again, a very generous grant from the NIH to build this next generation scanner, Connectome 2.0. And Randy, maybe you want to advance for me, since I realize I don't have control over the advance. You do. There, up, go back. There you go. Okay. So what are we talking about when we're talking about brain connectivity? You might imagine, we've been dissecting brains for, a very long time now, more than 100 years of, kind of rigorous scientific study. And yet, surprisingly, we know actually very little about how the brain is really wired. Like the very biggest wires and connections we have a sense for, but the tools to be able to image how the brain is connected really are extremely limited. And about 25 years ago, almost 30 years ago now, one of our colleagues here at the Martino Center, Van Wadeen, working with one of our graduate students in the MEM program, Tim Davis, came up with a clever way to use MRI scanners, not just to take kind of pictures of the anatomy of the brain, but actually show the wiring. And that picture in the center that you could see with the little white box along, that colored picture, is kind of one way that we represent how the wires are connected in the brain. We do this by measuring literally how water molecules diffuse, the random Brownian motion of water molecules. And it turns out that the water molecules like to wiggle along the direction of fiber axons in the white matter of the brain, and less so perpendicular to that. And so if we can measure the direction that individual water molecules diffuse or wiggle, we can actually make maps. And so these are just some pictures on the bottom. You can see kind of comparisons between the Connectome 2.0 and 1.0. And that kind of extra color, that extra detail in the Connectome 2.0 are basically going to allow us to kind of really understand in the human brain how things are wired. Randy, next slide. So so one of the real challenges when you do these experiments is that we've determined that while we know how to take these diffusion images, and amongst other things, we had to redesign the MRI scanner in a way to really be kind of maximally beneficial for this kind of diffusion imaging. And that involved optimizing the part of the MRI system, the gradient coil. That's the part that gives the knocking sound. If you've ever been in an MRI scanner and you know that kind of rattle that it gives, that comes from the gradient. That does our spatial encoding. It takes the picture. But it turns out that when you really kind of crank that gradient and its performance up to 11 to really take the most interesting biological pictures, that you begin to get some biological effects that are, really disadvantageous. In particular, your peripheral nerves can be stimulated. So you could design a system that could be great at imaging the brain, but that nobody would stay still in the scanner because they basically feel at first a tapping sound and then a tapping feel, and then that tapping becomes, more and more painful as you crank up the gradient. In other words, it's not so much the technology, but it's actually the biology that limits how we can perform this scanning. So two of our colleagues, Matthias Davids, a young scientist in the group working under the mentorship of Larry Wald, a senior physicist, actually developed a very detailed computational model of both the gradient and how that gradient actually stimulates individual nerves in the body. And they built a system that actually maps every nerve, peripheral nerve in the body, and then could project from any given gradient design how it would stimulate. And from that, they discovered that they could actually build optimization of that gradient design to minimize this biological effect. So this project has all these interesting elements. It has this interesting biology of how the brain is wired. It has this interesting kind of mechanical engineering and electrical engineering and biophysical modeling that we could use to try to reduce the biological limits to allow us to push the technology even further. A really interesting project in that way. And we had students, graduate students in our project at every step of the way. Next slide. So here's this kind of big picture of how we're imagining how we can use the Connectome. That's the kind of yellow box on the left-hand side as kind of an intermediate point as we want to work down to ever finer scales using optical coherence tomography, using clearance techniques, using micro CT imaging to basically bridge what we can see in a living human all the way down to measuring individual axons. As you can see, that light sheet microscope, that LSM picture, individual axons that can be labeled an image. And our goal is to try to build the connections between all of these different parts of the scale. And this involves not only folks at the Martino Center, but of course throughout the rest of Harvard community and throughout the rest of the country. And these are the people that are involved in that project down below. Next slide. Now, we're very big on MRI at the Martino Center. We used to even be named the MGH NMR Center back in the day. Nuclear magnetic resonance, of course, is the core technology for MRI, magnetic resonance imaging. But it's not the only thing we do. One of the other technologies we spend a lot of time working on, both using and now building new tools, is positron emission tomography, or PET scanning. This is an amazing tool because it allows us to look at individual molecules within the brain. And my colleague Chipper Katana, along with, again, a kind of a family of colleagues with expertise ranging from electrical engineering, computer science, physics, including a physics graduate student who's part of our NTP program, are building a brand new scanner. I think the next slide kind of highlights the different elements of it. You can see it's really a very unique design. This is not the typical tube that somebody would go in. It's actually a helmet that goes all the way around the head. That allows us to capture kind of like every single photon, or nearly every photon that goes around the head. We haven't quite figured out how to extend it around the neck. Most of our subjects are not very keen to have coils, and crystals where their necks are, but we can get a lot of coverage, and that actually allows us to improve our efficiency of collecting the signals that we get from a PET scanner by almost a factor of 10, an order of magnitude. Of course, the other challenge is we'd also like to have the MRI data at the same time. And so Cyprian is now building this very high resolution, unique design PET scanner to actually live inside an ultra high field MRI scanner. So we can really get the benefits that you just saw with the Connectome of these wonderful images of brain anatomy with the MRI, and yet do that with full kind of neurochemical sensitivity the way we can with PET. Next slide. And so much more. I think click through, we're talking about now, and in fact are now developing handheld MRI scanners that can go almost anywhere. Other kinds of imaging modalities, some of which you probably never heard of before, things like magnetic particle imaging. Larry Wald, the fellow I mentioned before, has actually developed the first human magnetic particle imager. It's just coming online now. We're doing brain stimulation and combining that with imaging methods. This is combined transcranial magnetic stimulation. That's the TMS part of that description, with optically pumped magnetometers, OPM MEG. MEG stands for magnetoencephalography, kind of the expensive magnetic cousin to EEG. We're combining those technologies. Portable imaging of blood flow quantification optically, which we're doing in little babies and actually doing now around the world, Maria Franceschini. So all sorts of things are happening within the program, and every one of these projects here and elsewhere has been worked on by graduate students within our program. And we're keen to see some of you come by and work on these projects with us. I think that's it, Randy. Am I correct? Okay, so now I guess we're popcorning over to Josh. Is that correct? Josh, you'll introduce yourself?
Yep, perfect. Thanks, Bruce. Awesome. So hi, everybody. My name is Josh. I am a student in the medical engineering medical physics program that Randy introduced. And working at the Martino Center as part of the neuroimaging training program. Out of the 3 students that will talk to you guys here today, I'm the newest of the group. So this is my first year in the program. And just last year, I was around this time finishing my senior year of undergraduate studies. So understand a little bit, I guess, how it feels in that stage. And I'd like to share some of my journey sort of leading up to this point, with the hope that maybe some of the experiences and lessons that I've learned could be of use to you, both in your undergraduate studies, in your future career plans, and beyond. So again, I'll start off a little bit talking about my undergraduate experiences, as you can see here in the top slide. So from a pretty young age, I knew I wanted to somehow contribute to medical research. Now I had absolutely no idea what a career in medical research looked like. I didn't really plan to be a practicing doctor. Had some family members with health challenges, and that kind of increased my motivation as well. But as I got older, I realized that instead of maybe biology or chemistry, I really like physics, and I really like physics and math, and so I sort of had this question in my mind as I started college, as you can see in red on the left, which is, how does physics fit into medical research? So I sort of began with that question in mind and started off at the University of Utah, having chosen physics as my undergraduate major. And after a couple of semesters, I wasn't sure that it was going to lead me to work in the space that I was most interested in medicine. And so I switched to biomedical engineering, BME, with the hopes of maybe, doing a 4-year and done degree in the biomedical space. And at this point, I had absolutely no plans to go to graduate school. But I realized after spending some time in biomedical engineering that I really missed physics. I missed deriving kind of physical laws from the fundamentals, the rigors of the math that come with that. And so after a lot of indecision, I switched back to physics, taking a leap of faith, sort of unsure where it would lead me. And I ended up adding a minor to that to use my biomedical engineering courses that I'd taken. So I got a minor in BME. And at that point, I said, well, maybe I'll end up doing a clinical medical physics career. And if you're not aware, a clinical medical physicist generally helps administer radiation therapy, or they might help manage imaging machines and protocols in a clinical space. They're often working with patients or doctors on a regular basis, and not necessarily doing a lot of research. So about 2 weeks after kind of making this switch once again to major in physics, I ended up speaking with some professors in a lab that worked on magnetic resonance guided focus ultrasound treatments and laser treatments in the brain. And I realized, speaking with them, that they were looking for a student specifically in the physics department. And so I jumped in, and I realized I had sort of found the answer to my question. I started discovering a new space where I could use physics to do medical research, and that was with imaging research and imaging as it applies to therapeutics. So these are some images up top of some things that I worked on there. So I did a lot of MRI temperature imaging, laser heat modeling. I also, through my research there, published a work on focus ultrasound simulations through skull bone models. And I worked in that lab for about 2.5 years before graduating. So I'd like to sort of pause for a moment and talk about 3 of the kind of big key lessons that I learned from that process, and that hopefully will be helpful for you guys as esteemed scholars and wherever you are in your path in your undergraduate studies. So the first one of those lessons that I wanted to talk about was it's worth pursuing what you're really interested in. So even if you don't know exactly what your job will look like in 5 years, 10 years, I think in many cases the right doors will open if you are passionate about what you're doing. So in my case, that was sort of this combination of physics and medicine. In your case, that might be biology or chemistry or a branch of engineering. But I would encourage you to find what you enjoy and to go for it. And something that I really have appreciated about where I'm at currently is the breadth of opportunities that are available both in the MEM program, and then also doing research within this NTP program. There's space for really all of those disciplines. So pursue what you enjoy, and you never know what opportunities are out there for you to find. Kind of the second big lesson that I learned from this process was that getting involved in research is very helpful to kind of teach you if you like the research process. I think it's very important, as was mentioned earlier on, very important to have some research experiences before grad school, and that could be a summer research experience, that could be a year or multiple years of research experience. But even more than just sort of like the scientific questions, I think it's helpful to understand if you like the opportunities and the challenges and the type of work that comes with research. What does doing research as a student, graduate student, postdoc or a professor look like, sort of at each of those stages, and what work am I interested in doing now and in the future. And sort of going through this process of not really being aware of a lot of those answers, learning as I went, was very helpful for me. And the third lesson that I would sort of emphasize was to find good mentors. So my advisor in this process, Dr. Dennis Parker at the U of Utah, was important in kind of finally convincing me that research was a possible career plan and could be a good choice for me. For a while, even while working in the lab, I was still partially convinced that I needed to find a different career route. And I think it was just, I wasn't aware, I think, of of what that path looked like and what opportunities it could bring. So having those experiences and having, good mentors who are aware of your strengths, your weaknesses, and your interests to help guide you, I think is very important. So as of this year, as I mentioned, I'm now here in the MEM program at joint between Harvard and MIT. So shown in the bottom slide. And I was very excited about the program specifically because, as Bruce mentioned, I can continue to sort of dive deep into the fundamentals, the science and engineering here at MIT, and then also, the medical education through Harvard Medical School. So just briefly kind of going through my current research interests, plan for my PhD. So sort of following in suit of my past experiences, I'm very interested in using MRI to measure various biophysical properties, including blood and fluid flow, diffusion, microstructure. You can see here in this image in the middle on the bottom, these images on the bottom are not mine, but from some related papers and colleagues here locally. So this middle image shows multi-contrast MRI looking at things like diffusion and blood flow and permeability of vessels within the brain. The other component of my research interest has to do with sort of the biophysical modeling side. So you can see this image on the right that shows a vascular model that can be used to model things like drug delivery in a region of tissue. So sort of tying those together, my thesis project plan has to do with using MRI and other types of imaging to model drug delivery in brain cancer. And I'm currently working under the direction of Bruce, who just spoke with us, and working with other collaborators. So feel free to reach out over email or in the chat if you have other questions, or if there's any way that I can help support you in this process. I know there's a lot of questions, a lot of decisions to be made along the path. So I'm happy to be a support and a contact for you as needed. And with that, I will pass the baton over to Amelia. So.
Thanks, Josh. Yeah, hi everyone. My name is Amelia. I'm a current third-year MEM. I'm co-advised currently by Jonathan Palomini and Laura Lewis, again at the MGH Martino Center. And just really quickly about the research that I'm working on now. So my thesis project is focused on the mechanisms of CSF flow in the brain, cerebral spinal fluid flow that's involved in waste clearance and aging, and really important for development of neurodegenerative disease. And specifically, I'm using 7 Tesla MRI to quantify brain tissue motion. So like the brain tissue is quite soft, so any mechanical inputs like the heartbeat actually induce some displacement, and that might be involved in directing CSF flow. So I look at a lot of pictures like that on the bottom in the middle of these phase valued MR images to quantify motion. So these are different phases of the cardiac cycle, and I'm really enjoying it. So just as a quick outline of my path here is a bit longer than at least most folks in my cohort. Since I didn't enter grad school directly from undergrad, I took a few years off in between. So I'll share my decision-making there and sort of how I ended up here, and hopefully there's some things that can help you all out. So I did my undergraduate degree at UC Santa Barbara, majored in biopsychology, which is, at least at UCSB, it was a kind of a psychology degree with pre-med requirements. And when I started, even as a freshman, I knew I was interested in brain science. I knew I was interested in research. But I didn't actually do that much research when I was in undergrad. I actually spent my entire junior year studying abroad, so I definitely didn't do any research at that point. But in my sophomore year, I ran some data collection for an effective psychology lab. And then my senior year, I really buckled down and completed an honors thesis in an EEG lab, where again, I was leading data collection and wrote that up for a thesis. And by the time I graduated, I knew that I enjoyed the research process, you know, even with those sort of few experiences that I had. And I knew I would probably want to go to graduate school eventually, but I didn't have nearly enough research experience to like know what kind of research I actually wanted to do for my PhD, and two, to like be a competitive candidate for like a top program like HS or like the MEMP program. At UCSB, the focus is kind of on cognitive neuroscience and like psychology, and I just didn't find something that I gravitated towards at UCSB. And also, they didn't send too many students to graduate schools like MIT. So I remember even like an academic advisor telling us, like, I know a lot of you want to go to grad school, but many of you aren't going to get accepted and you should lower your expectations. So by the time I graduated, I didn't think like grad school was the right choice for me, like right then. So instead, I wanted to find something I was more interested in. I knew I liked clinical sort of applications and more of a biology, physiology focus. So I got a job working as a research assistant at UC San Francisco in a dementia lab that used PET imaging. So like Bruce mentioned earlier, we were looking at proteins amyloid and tau in the brains of patients with various dementias. And I stayed there for 3 years. And that's really where everything came together for me for a few reasons. For one, the research was a lot more interesting and enjoyable for me. So we were working with clinical populations. It felt really meaningful and motivating. And I knew at that point that I really enjoyed biomedical imaging research. And I was also able to get a lot more experience with other steps in the research process. I didn't do much data analysis in undergrad, and that was the core of my role at UCSF. So I really got a lot more experience there. And I also was able to work on my own independent research project that culminated in my first author paper. That was a really valuable experience. And then maybe most importantly, I was incredibly lucky to have fantastic mentors there who really invested in my professional development and in my technical growth. So they encouraged me, even just being able to set aside time to complete an entire independent project, I think speaks a lot to their investment in me. And then finally, it was really valuable for me to spend time doing research full time to ensure that, I really enjoyed this process and I really wanted to do it as a career. So really valuable. So by the time I was applying to PhD programs, I was in a much better position. I had figured out what kind of research I wanted to pursue, through talking with colleagues and working with different labs. I was really interested in brain physiology and like the vasculature, how all that contributes to the neurodegenerative process. And I became really interested in MRI and the versatility of it, especially for clinical practice. So I decided to pursue PhD programs to really do this neuroimaging with MRI with a clinical focus. And then also by the end of my time at UCSF, I was a much more competitive candidate. I had my name on papers, things like that, and had this wealth of research experience to talk about. Now, when I started at HST, at MEMP, it's such a valuable opportunity, especially with the neuroimaging training program, to get involved with methods development and really learning about, developing an MRI method, and get involved in biomedical imaging. And at that time, I had no business doing any sort of engineering. I had my degree, my experience at that point was basically purely in the biological sciences. The last math class I took was in high school. I really had to buckle down there. So I had to do a lot of independent learning my first year in the program to get prepped for the MIT coursework. So I took signal processing and linear algebra online. read textbooks on MRI physics to gain an intuition into that. And then, of course, the community in the MEMP program and in the neuroimaging training program is fantastic, and it's very engineering focused. So I had a lot of friends that I could lean on to help me get up to speed, so to speak. And some of them are shown in that photo there. So when I was looking for a lab, I knew from UCSF that I wanted to find a fantastic mentor. And to me, that means somebody that invests in you and your goals and what you want to develop as a scientist, as a researcher. And I also wanted to find someone that I could learn method development. So I really wanted to have like 1 foot in the development, 1 foot in the application, and really be able to speak both those languages. And I chose my current lab, mainly working with John Polemini for those reasons. So I guess at the end of the day, maybe some themes that we can take away, really similar to what Josh was saying. So for one, for me, I would say that it's okay to not have these like amazing research experiences in college. If you don't find something that you gravitate towards, that you're passionate about during your undergrad years, like take your time, don't force something that's not enjoyable for you. It's not going to make the PhD any easier. And then next thing is finding great mentors. And again, for me, what that means is people who invest in you and really work with you the way that will best meet your goals. And finally, the last thing I'd say is to really know the gaps in your knowledge and take the initiative to fill them. So I like I like the saying of stay on the steepest part of the learning curve. That's certainly something that I've done in the roles that I've taken on, and you'd be surprised at what you can do. So yeah, like Josh said, if you have any questions, if this piqued any interest for you, happy to answer any questions here or via email. I'll turn it over to Vivek.
Cool, thanks. Hi, everyone. I don't have a slide, so I'll just like talk. But I'm same as Amelia. I'm a 3rd year in the MEMP program. I'm a little less focused on neuroscience the same way that Josh and Amelia are. My interest in neuro is much more from like a surgical point of view. So my specific focus area in MEMP is specific within computer science. And like one of like the side passions that I picked up when starting my PhD was actually in computer graphics. So what I really work on now is building like 3D image guided systems so that neurosurgeons can do surgery easier. I didn't make a slide, but like I'll put one of the things that I've made just in chat. It probably gives a better overview than I could give of like what I actually work on. But like I just very much like have come to enjoy like all of these things that you hear about when people like build video games, but basically applying that to medical images. So like the thing that I just linked is a ray tracer that will basically simulate X-rays from 3D CT scans. And all of my work is basically around exploiting all the information that we have in X-rays that you might use during an actual surgery and trying to create more informative ways of visualizing anatomy for surgeons. But yeah, that's basically my research. I'm happy to talk more about that if people want. But what I thought might be a little bit more useful is like same as Josh and Amelia to talk more about like my journey to how I got here, and kind of more about like what I learned along the way and what I wish I had known earlier. One cool thing is that my roommate, who's also in this MEMP program, was an esteemed scholar. He did his undergrad at UT San Antonio. So like I took the time to like, we talked just a bit about like what he wished he had known as well. So this is a bit of like a 2-for-1, like it's like half of his input as well. But I think I'll pick up where Amelia left off on mentors. I think like the mentors that you find in undergrad are incredibly important for like giving you the confidence to pursue research at any level. But in undergrad, I was really, really lucky that I found someone who really cared about mentoring undergrads. Every year in his lab, there would be like 10 or 15 undergrads that rotated through. And like, he didn't do all of it, right? Like he delegated properly. So PhD students and postdocs were like doing day-to-day, like working with us, but he really cared about mentoring undergrads and like getting us to a point where we really could do computational research. One of the things that I really liked about him is he was the most blunt person that I've ever met. If I was doing something wrong or he didn't like how a figure looked or I did like an analysis wrong, like he would just tell me in front of everyone. And I know like that doesn't work for all people. Like you need to match who your mentorship is or who your mentor is with what kind of style you like. But for me personally, I really like that style of mentorship and I grew really rapidly working with him. And so it was less about like the curiosity of research and like what I was really interested in exploring, like all those things I thought I'll figure out later because I found a mentor who really taught me the nuts and bolts of how to do research, and I really loved working with him and his team. So definitely finding a mentor who cares about mentoring undergrads in the first place, I think is really critical. There's a big difference between mentoring grad students and mentoring undergrads. So finding people who really have a track record for mentoring undergrads and not just like using them as grunts to do like tissue culture, I think is really important when you're looking at it from an undergrad point of view. The second thing that I want to highlight is once you find a good mentor, I think it's like the luckiest thing in the world, like someone who really cares about like mentoring you and making you like the best possible research that you can be. Like that is so special and it is really quite rare. So when I was talking with my friend who was an esteemed scholar, he said, yeah, once he found that person in his undergrad, he was like, it was such a joy to work with her that he's like, all right, I'm ready to do like 20-hour weeks in lab. This is like kind of the part about like one of the truths about research, which is just like it's a really like it's like almost like a sport, like you need to practice. And so a lot of it is just like really getting in there, doing whatever project it is you're on, really getting invested in it and like just kind of thinking about it. It's just about like sitting and like marinating yourself in that field, and suddenly like ideas will come to you when you least expect it. So just like really being like excited, like you should find something that is so cool to you that it's like I'm excited to like grind on this, even if it means like I'm not going to go to this party or like, oh man, maybe I'm not paying attention in class. It's like, you know, for every person will be a little different, but this is kind of like how I felt at some points in class. Like if the lecture wasn't interesting, I was pulling out my laptop and looking at research because I just couldn't like not look at it for that hour. It's just like the bug just kind of bit me. And then I think the final thing that I really, really learned in undergrad research that is probably the most translatable thing to what I do now as a grad student is really learning how to teach yourself something. Like there are great PhD students, there's great postdocs, there's great PIs, but like they can't be with you 24/7. Like if you get stuck, one of the most useful skills you can learn is how to unstick yourself. And so from a coding point of view, this was like really knowing like if I hit a bug in my code, how to debug it, how to figure out what the solution is and move forward. But I think it really becomes a little different in grad school. Amelia was alluding to this earlier, but it's kind of like how to teach yourself entire subjects, not necessarily at like some great depth, but like at a cursory level where you can start to make inroads, where you can like figure out what exactly it is you don't know. So like for me, I had a really super heavy engineering background in undergrad, but like the converse of Amelia's thing, like I hadn't taken a bio class since high school. So like it wasn't until I was in the neuroscience class at Harvard and MIT that like I figured out what the difference between an axon and a dendrite actually was. Like, that's kind of embarrassing to admit, but like you just really have to know how to address your own holes in your knowledge and kind of be unashamed of the fact that there are going to be so many and just like enjoy plugging them up. But yeah, learning how to teach yourself things that you don't know is probably the most valuable skill that I learned in undergrad. And it's the thing that has carried me forward the most in PhDs, especially like as you like in a PhD, you find things that you didn't even know that you could be interested in. And you're like, oh, that's really cool. I kind of want to wade into that. So knowing how to teach yourself that foundational like background knowledge is really, really helpful. Yeah, I didn't make a slide, so you don't have my email. I'll just put it in chat. Happy to like answer any follow-up questions either now or over email about that stuff. But yeah, thanks.
Thank you, Vivek. Thank you, Amelia. Thank you, Josh. Thank you, Bruce. When I put out a call for students to present, I actually was blown away. Within like an hour, I had 6 applicants, like that, way more than we would have time for. So if you want to hear a little bit more from a few more people, I've passed along to Khalil their names and their interests and emails so that he has a few more student ambassadors. So there's, I think, 6 total people you can contact if you have questions. But
So
does anybody else have?
I've got a question. All three of you have really focused on the importance of the mentorship. And you've described some of the characteristics of a good mentor that you've all been fortunate to kind of connect to. But how do you actually find that person? Like, what are the tricks to finding the mentor? Like, okay, these are the characteristics, and once you see it, maybe you'll know it if you see it. But how do you find that person? Anybody?
Sure, I can jump in. I think the first thing that I did was I looked at my department's faculty list. I crossed off like the 80% of people who didn't work on stuff that I was interested in. And then like I boiled it down to maybe like 5 people whose work I thought was interesting. And then what I did is I found like very new grad students in that lab, or even like senior undergrad. So like I started doing research when I was a sophomore in undergrad. So like I found seniors in the lab who I knew, like just talked to them one-on-one and figured out like what was the style like. And that's when I found out that this guy that I ended up working with for the next 3 years had this like group of 15 undergrads who cycled through every year. It's okay, probably seems like a good mentor.
I would like to add, I think for many undergraduates, we recommend NIH-funded National Research Mentoring Network Program, NRMN, where that platform has got opportunities to choose a mentor or choose a mentee. I strongly suggest I make it a mandatory for all my students to enroll into that. Before even they go to the grad school, they can choose a mentor and then pursue. They need not go to the same institution, but that mentor can put them in the right network. So that's what that platform is very effective right now. I believe they have more than 10,000 both mentors and mentees. I think I mentioned that one to Tina last time when I gave the presentation. It is there. It's a very well established platform to choose a mentor for, especially for all levels. It need not be just undergraduate, for graduate student, postdocs, early career scientists or faculty. It is there. It is there. It has been there for 12, 13 years. I have been part of that, and it is managed by University of North Texas Health Science Center.
I'll give, one other, kind of twist on that from my own experience, which is don't be afraid to move on. If you find somebody that you think is the right one and it turns out it's not, move on to the next one, right? I mean, I went through 3 different mentors, 3 different projects as a grad student before I found the right one. And when I found it, it was great. And, 40 years later, I'm still, in that same field. But the first couple, I mean, great people, they weren't bad people. And it's not even that they were bad mentors, but the combination of what they had to offer and teach me, and the nature of the project and my vision for where the project was going just didn't all mesh to fit with me. And so don't feel like if your first, mentor is just not working out, that you have to stay there the rest of your career. Keep looking. It's not so bad to get a couple false starts to find the right one.
I'll chime in with the concept that mentoring is also not a solo activity, it's a team sport. Because a lot of times it's hard to find. I never found the perfect mentor ever. And for me, what ended up working really, really well was having several people who, each of whom contributed something super valuable and super helpful and were awesome. And you just have to put together a network that works, a network of mentors that will work for you if you're not lucky enough to find yourself in the spot where all those things line up the way in a perfect world it would be nice if they do, because they don't always.
Can I ask a question?
Go for it.
Can everybody hear me? There's some background music here in the Starbucks, but apologies in advance. Well, first of all, I have a comment. One, I'm just blown away by this program. I wish this was available to me when I was in high school or college. It's amazing. 2, I'm blown away by these 3 students who have shared their work with us. If I was at MIT and I had a contract that I could force them to sign to get them on the faculty, whenever they graduate, I would have the paperwork ready right now. You all are brilliant, and you clearly have a direction and a path that you all are intensely pursuing, and I hope that you keep going. I guess my question is, and this is a challenge I have here at the University of Colorado, working with community college students who are underserved and underrepresented, in trying to get them to kind of figure out probably what you all may have figured out quite easily years ago, what they really want to do. And when I was in graduate school, and I'm dating myself here, I knew that I could do medicine, nursing, public health, and some type of research. And that was kind of like my lens. Neuroimaging, on the other hand, is not only kind of a niche within medical research, but it's also a niche within imaging. So I would imagine that a lot of high school and college students, and maybe things are different now, but I would imagine many of them, if asked to define what is neuroimaging, maybe a lot of even science-oriented people couldn't do it because it's so focused. So I'm curious, like, how did you all, or when did you all discover, like, okay, neuroimaging is what I really want to focus on and what I want to do, as opposed to, I don't know, something more, maybe more generic, like pediatrics, which is what I did. And even within pediatrics, for example, there's cardiovascular pediatrics, there's pulmonary pediatrics, there's infectious disease pediatrics. To me, it feels like neuroimaging is almost like something you have to know that you wanted to do, maybe after the first physics or calculus class that you got an A+ in and said, okay, or maybe had a parent that did it, or an uncle or an aunt that did it and took you to their lab when you were 7 or something and kind of got you in that direction. But I'm kind of curious, both to the students and to the mentors, like, how do you kind of expose young people to kind of a niche area of research and also package it in a way that doesn't intimidate them and overwhelm them thinking, oh, well, I have to know neurology, I have to know physics, I have to know math, I have to know medicine in order to pursue this goal, and also make it like attractive enough for them so that they can understand, okay, well, maybe it can be as glamorous as brain surgery or bioengineering, like, I'm sorry that was a loaded question. I know that was a lot, but I'm just curious, like, how you kind of get young people to even approach this arena, if that makes sense.
I think we would all be able to answer that when you start, Josh.
I was just going to share from a student's perspective. I sort of mentioned that, I mean, I kind of knew like what general stuff I liked, but I really wasn't aware of like the details of the field, sort of as you mentioned, Dr. Walker. And so to me, that like sort of eye-opening moment came, it was actually like a kind of a seminar style class that was part of the program that I was at in undergraduate, that they had to come in and just do short, like 5 or 10 minute presentations on what they were working on. And I think, trying to kind of disseminate research in a way that can be grasped, according to the audience. At least for me, it was effective enough that I was interested in what the lab was doing. That's why I applied. And then, stepping into it, I sort of learned more about like, the world of, I guess, brain MRI, like as you mentioned. And so, if there's opportunities to communicate and share, core research progress in a way that's digestible out of high school or an undergraduate level, I think those are helpful opportunities. So.
Yeah, maybe I'll add to that. From my perspective, like I came in knowing the neuro part. I wanted to do something with that neuro part, but the imaging, I think it's like a lot of folks, especially, you know, at least in sort of brain science-esque areas, you get exposed to some of these things. Like I remember in class learning about the default mode network, and it blew my mind when I was an undergrad. And it's those sort of tidbits, you know, there's like certain pieces that you learn about in classes or something like that, that just like sort of are earworm in your brain, like, wow, that was really interesting. I want to get into more of that. So I think it's two things on the side of the educators to include all these different niches in the coursework, and then on the side of the student to recognize which of those is really like, you know, piquing your interest, and then dive into each of those as they come up.
I will say I mean, obviously neuroimaging is a niche and it's not going to be for everyone. It does have, you know, certain things going for it. Most people are interested in the brain. They're interested in their own brains, how brains work, right? I mean, even if you know, you may have interest in all sorts of other things, but the brain is kind of naturally kind of an interesting organ. And of course, you know, we're so far away from understanding it that it's easy to imagine, you know, spending an entire career, you know, and your children's career and your grandchildren's career trying to understand it. And then the imaging part, well, I mean, of course, there are lots of ways to do neuroscience, you know, in test tubes and in plates and in individual neurons. But if you want to study the human brain, you know, especially like the living, acting human brain, well, that's neuroimaging. So if you're kind of cool on the brain and you actually are interested in how the human brain works, that does kind of naturally lead you into this direction. So it's a niche, but not such a tiny one. And it's usually not too hard, you know, at the high school level and up to get people interested in some facet of how their brains work. And then if you can put them in a scanner and give them a picture of their brain, that gets everybody juiced.
Yep. For me, there is
Thank
nothing
you.
else. So Zeynep, do you need anything? So thank you for having us. We really appreciated the chance to share what we were up to. And thank you all for being such a lovely audience. And, you know, it's nice to spend an afternoon with you.
Bruce, Josh, Amelia, Vivek in the order of appearance. Thank you so much for making time for us. And for the attendees, I want to remind you, please send any feedback. Khalil popped the survey form in the chat. If you could go there and give us any ideas to improve, especially maybe how to recruit more people, like if you know about some of your friends in college that would have come but couldn't come because of a certain reason, if you could give us any ideas about how to improve. And the other point I wanted to make is that, like Randy mentioned, there's a list that has the addresses of our three speakers today in the student side of things, and also a few others. So if you are interested in receiving that, you could write to Khalil, and Khalil also put his email in, you know, in the chat closer to the top there if you want to take that down. So thank you very much, both to our speakers and to our attendees, and keep well.
Thanks for the opportunity, Zainab. Great to meet you all. Hopefully we'll see you in Boston.
Yes.
Thank you.
Bye-bye.